Hello and welcome to Cyclica's AI Ate My Homework. This is a 7-part webinar series that will provide key perspectives in science, technology, entrepreneurship, and the exciting field of AI and drug discovery. It's great to see an eager group of participants with diverse education levels and areas of expertise sign up. We will begin the series by covering foundational topics, and as we progress, we'll introduce more advanced topics on machine learning and chemical biology. Be sure to follow us on LinkedIn and Twitter, where we'll be announcing new topics on a weekly basis. If you have questions, please use the Q&A feature in this webinar, as questions will be answered toward the end of the session if time permits. My name is David Chauhan, and it's a pleasure to introduce our panelists today, Ben DiFrancesco and Shelley Deeke, who will be discussing the impact of biology on health and to develop medicines.
Thanks, Devesh, for the introduction. My name is Ben DiFrancesco, and I'm just going to give a very brief introduction on sort of my scientific background and my, I guess, path to where I am now. So I did a Bachelor of Science in Chemical Biology at McMaster University in Hamilton. And there, while I was doing a fourth-year project on the synthesis of drugs to treat breast cancer, that sort of triggered my interest in the chemistry and biology of drug discovery. So from there, I did a PhD in biological chemistry at U of T, and I'm going to talk a little bit about the work I did there. So the work I did surrounded bacterial biofilms. And so when traditionally you think of bacteria, you think of them kind of swimming around and floating as these little independent cells, but that's actually only really half the truth. So really when bacteria are infecting anything, what they're doing actually is adhering to surfaces. And when they do that in this biofilm state, they actually create this sort of shield that protects them from antibiotics, it protects them from your immune system, and it can protect them from any kind of attempt to scrape them off. So what I did during my PhD was to try and understand first the protein machinery that was used to build this, this shield, I suppose. And what we found is that these shields are actually made of big long chains of sugars that are built in the bacteria and exported and modified to form that shield-like layer. And from there, we looked at the proteins that built up this molecular machinery and studying their structure and their function. Once we had understood their structure and function, we could look at them to develop drugs that bind to them in order to prevent the production of these sugars and thereby prevent the formation of these biofilms. And when we had identified and discovered sort of new ways of inhibiting these proteins, I went into the lab and I was able to use organic synthesis to build these molecules and test them against these proteins and put them back into these bacteria to test to see whether they would inhibit bacterial biofilm formation or not. From that experience and of trying to discover new molecules, I was really intrigued on the power of using computers to accelerate that process. And that's what led me to where I am now. I'm an applied scientist at Cyclica.
Great. Hi, everyone. My name is Shelley Deeke. Let me begin by providing you with a little bit of background on myself. So I also work as an applied scientist at Cyclica, which means that I work with our partners in applying our technology to help them reach their research objectives in drug discovery. So after my master's in biochemistry at the University of Ottawa, I was working as a research assistant at the Ottawa Institute of Systems Biology. And I really fell in love with the proposed research project, which was in collaboration with the Children's Hospital of Eastern Ontario, which is why I chose to pursue my PhD. And so the research project was really at the interface of fundamental science and also the clinic, which is what really resonated with me. And so part of my PhD project was to find better ways of diagnosing children that have inflammatory bowel disease. So this includes both Crohn's disease and ulcerative colitis. Here I've depicted an example of ulcerative colitis where the entire colon is inflamed. And when this is the case, this is actually referred to as pancolitis. And so currently diagnosis relies on colonoscopy, which is obviously very invasive. And so one of our objectives was to try to identify biomarkers which would make this diagnosis procedure less invasive. And in order to do this, we applied proteomics, which essentially means a large-scale study of proteins. And on the right-hand side here of the slide, I just have some examples of proteins which show differences in expression between healthy children, which are shown by these blue circles, compared to children with IBD, which are represented by these red circles. And so in today's session, I'll be talking about proteins and how they are involved in disease. So let's begin with a zoomed out lens of the human body. So the human body, it's a complex system, and it's the coordination of all of its smaller parts working together is what keeps us alive. So our bodies contain different organ systems, and each of these have different roles. Organs can then be further broken down into cells, and to put this in perspective, humans are made up of trillions of cells. Cells can then be further broken down into organelles, which are small cellular structures that perform specific functions within a cell. So here I have an example of a particular organelle, which is mitochondria. And so these are often thought of as the powerhouse of cells because one of their main functions is to generate energy. And finally, we have proteins, which are the basic components of living cells. And notably, proteins are just one of the molecular components of organelles. So let's dive a little bit deeper into proteins. So the building blocks of proteins are called amino acids, and the structure and function of our bodies depends on proteins. So there's many different types of proteins with different functions. So for example, there's structural proteins. These allow our cells to maintain their shape. Some proteins are digesters. For example, pepsin helps digest the proteins that are in our food. Some proteins can be defenders. An example of this is an antibody, which allows your body to fight invaders. Some proteins are messengers. So for example, a kinase, which can in turn affect the activity of many other proteins. And for humans specifically, there are about 20,000 proteins which are coded by our genes. So now let's look at some more detailed examples of proteins. So let's begin with salivary amylase. So as its name suggests, it's present in your saliva, and it breaks down carbohydrates into smaller molecules like sugars. So this protein helps your body to digest starchy food like potatoes, rice, or pasta. So we've already briefly mentioned pepsin. So this is an enzyme which helps digest the proteins in your food. Another example of a protein is cytochrome P450. So these are enzymes that are mainly found in your liver cells. They metabolize external substances such as medications, drugs that you may ingest, but they can also metabolize internal substances, for example, toxins that could be formed within cells. And another example is antibodies. So these are used by our immune system, and they allow the neutralization of pathogens, for example, pathogenic bacteria or viruses. So let's move forward with what proteins actually look like. So as we briefly mentioned, amino acids are the building blocks of proteins. So there are 20 different standard amino acids. Here we have 5 of them illustrated, which includes alanine, phenylalanine, threonine, aspartic acid, and histidine. Now there are certain parts, which you may have noticed, of amino acids which are common to all the amino acids. So for example, we have the amino group here on the left-hand side, which you'll notice on all amino acids. We also have the acidic carboxyl group, which is on the right-hand side. Again, you'll notice this on every single one of these amino acids. Each amino acid also has a unique group, which is called a side chain. And so this is shown on the lower half. And what you'll see is that these are different between each of these 5 different amino acids. So proteins also have different degrees of structure. So the first degree is the primary structure. And this is the order of the amino acids. And these amino acids are linked through peptide bonds. The second layer of structure has 3D structure elements. And so some of the most common secondary structures are alpha helices and beta sheets. Following that, there's the tertiary structure, which is the 3D arrangement of single chain. And finally, we have the quaternary structure, which is the 3D arrangement of multiple chains. So as we previously saw, there are many different types of proteins and they execute different functions. So let's take a look at enzymes, which perform all of the basic chemical tasks needed to sustain life. So enzymes act as a catalyst in biochemical reactions. What this means is that they speed up the reactions. So here we have a substrate, and a substrate is what we call a molecule upon which an enzyme will act. And an enzyme can act upon one or more substrates. So the enzyme will bind to its substrate target molecule, and it holds it in such a way that the chemical bond breaking and bond forming processes can take place more readily. And so the region where the substrate molecule binds on the enzyme is called the active site. So the enzyme will then perform a chemical change, and then it releases this altered molecule. So a few slides earlier, we talked briefly about salivary amylase. And this would be an example of an enzyme which is found in your body. It would break down amylose into its smaller sugars. So here previously we just saw an example of an enzyme, but as I mentioned, proteins can perform a variety of different functions. And so here's an example of how some proteins can actually act as a message. So our cells in our body are constantly in communication with each other. They send each other a variety of different messages. For example, they could send messages to divide or that there's dangers that are present. And so proteins can actually act as this signal where they move between cells like a message. So this is shown here as this purple circle, which is meant to depict a protein which has been released by this cell on the left-hand side. And so this protein is released and it can bind to a receptor on another cell. And the receptor here is shown in green. And once this protein binds, this can turn on the receptor. So once this receptor is turned on, what it can then do is initiate a signaling cascade. So what this means is that it's actually forwarding this message to proteins inside the cell so that the cell can then go on to take the necessary action in order for it to be able to divide. So here's an example of when things go wrong. So for example, there's receptors that are present on normal cells, but sometimes there are mutations in a gene. And what this can lead to is that there are too many of these receptors that are present on certain cells. And what this leads to is that there are more signals that are telling the cell to grow. So for example, here we have a first signaling cascade, which is launched, which is indicating to the cell to divide. Then there's a second signaling cascade that is launched, again, leading to another signal for the cell to divide and another round of cell division. So there are more and more signals that are telling cells to grow rapidly, and this can cause cancer. So here's an example of how targeting certain proteins can actually treat disease. So drugs can help restore this balance. So drugs can block the ligand from binding to its receptor. So here in this diagram, the happy faces are actually meant to depict drugs, and by binding to this receptor, they're actually preventing the signal from being turned on, and therefore it's preventing the cell from dividing, as the cell will never receive the message for the cell to divide. So this can prevent the uncontrolled cell division, which can be seen in cancer. And so I hope this example and what we've talked about here sort of demonstrates how biology can help us guide the development of new drugs, because understanding how processes work in the body can provide us with strategies on, for example, how to restore that balance. So in the example that we looked at today, we shut down the function of a particular protein, which was causing uncontrollable cell death in an attempt to treat cancer. And with that, I thank you for your attention, and I will pass it on to my colleague, Ben.
Thanks a lot, Shelley. Let me just share my screen. All right. So Shelley's done a really great job of explaining sort of how proteins come into play to cause and affect disease. And so now what I want to do and explain in this sort of half of the presentation is, how do drugs really exert this effect that Shelley just did a great job of introducing? How do drugs work? So I'm going to start in a very similar way to Shelley did in her section, kind of looking at the top down. And how I want to start off is just this example of, say you have the flu and your doctor prescribes you an antiviral. So what's going on there? So first of all, you want to remove your symptoms. So, cough or runny nose or all these sort of symptomatic effects. But why is that happening? So it's probably because you're reducing sinus inflammation or fluid buildup in your lungs. But why is it doing that? So the drug is somehow preventing your cells from dying and preventing them from sort of releasing inflammation signals to your other cells. But how is it doing that? And the answer is that it's interacting very specifically with a protein on the virus to destroy the pathogen and stop disease. So the question that I want to answer in sort of my half of this presentation is, how do drugs interact specifically in proteins, and how do we design them to interact specifically? How do we use drugs to affect human health? So to understand that, you really have to think about interactions between molecules, because as I've said on the slide, everything is chemistry, including proteins. And Shelley's already introduced that by telling us that proteins are just chains of amino acids strung together. And to that end, there's a number of different ways that you can look at a protein. So you can look at it as let me just turn on my pointer. You can look at it as this sort of blob, or sort of just the surface of the protein. You can also look at it in this form, where it's sort of these cartoon ribbons. And those represent just tracing the backbone of the amino acid chains. You can also look at it through this representation here, which is basically the chemical structure of every single amino acid in the protein. Similarly, you can look at small molecules that way, or you can look at drugs that way. And this is a structure of the drug penicillin, which is not to scale with the structures of the proteins over here. But here on the right, you can see this 3D structure of the molecule, where the carbon atoms are in green, you have the sulfur atom in yellow, and oxygen atoms in red. You can also look at the structures of drugs in this other way. And to introduce that, you may have heard humans described as carbon-based life forms, and that's very true. Carbon is an element that's present in our bodies and in biology in great numbers. And because of that, organic chemists get pretty lazy and don't want to draw the letter C for carbon over and over and over again. So in this structure, whenever you see a point or a corner, that actually represents a carbon atom. So this is a nitrogen bonded to a carbon bonded to an oxygen. And this ring here, which is a benzene ring, so this is actually 6 carbons all bonded together in a ring. So when I say that we want to talk about the interactions between drugs and proteins, what we're really talking about is the interactions between molecules, because a protein is just a big molecule. But to really understand what's going on, we actually have to go a level deeper than that. So as you're probably aware, molecules are actually built up of individual atoms. So to answer the question, how do drugs interact with proteins, we have to answer the question, how do atoms interact with each other? And the answer to that is that there's 2 major ways that they do that. So the first is called covalent bonds. And these are intramolecular bonds, means that they occur between atoms that are in the same molecule. And the way we represent that, for example, in our structure of penicillin, is just by solid lines. So in this structure, every line that you see is a covalent bond. And that's also true in this 3D representation here. Every single sort of line is a covalent bond between two atoms. But what we're really interested in is the interaction between drugs and proteins. So those are two different molecules. So that means we need to look at intermolecular interactions. And those are creatively called non-covalent interactions. And these occur in 3 different flavors. So the first is ionic interactions, hydrogen bonds, and van der Waals forces. I'm going to go over those briefly. So the first type of interaction, ionic bonds, occurs between ions. And an ion is simply a charged atom. And charges are either positive or negative. These behave very much like magnets, where if you have a south pole and a south pole, and you try and put them together, they're going to repel each other. But if you have a south pole and a north pole, those are going to actually attract. So in the same way, if you have 2 of the same charge, so a positive and a positive or a negative and a negative, those will repel. But if you have 2 different charges, like a positive and a negative, those will attract each other. And those both are ionic interactions. The second type of non-covalent interaction that I'm going to talk about is hydrogen bonds. And these are a very specific format. So here you have a non-carbon atom bonded to a hydrogen, and that hydrogen will hydrogen bond with another non-carbon atom. They occur this way, and it's not carbon, because of the way these atoms hold onto their electrons. And that causes a sort of very specific charge distribution that causes these hydrogen bonds to form. The last force that I'm going to talk about, the last intermolecular interaction, are van der Waals forces. So these are a little bit different than the other ones, where they're actually quite weak, and they occur in molecules that are primarily composed of carbon and hydrogen, or hydrocarbons. So if you have 2 atoms that are interacting with van der Waals forces just by themselves, that's actually not a strong interaction. But the interesting things about these van der Waals forces is that they are additive. So if you have a lot of atoms interacting with each other at the same time, these forces can actually be quite strong. An interesting way to think about this is that a lot of molecules or chemicals that solely contain carbon and hydrogen, or hydrocarbons, are oils and fats and grease. So to think of these as kind of greasy interactions in drug design wouldn't be far off. So now that we've sort of introduced these intermolecular interactions, let's look at how they apply to biology. And to start off, let's look at a system that's familiar, and that's DNA. So many of you, I'm sure, are aware of the genetic code being made of A, T, C, G. And it's very important to the function of DNA and thus of the propagation of life that this code can be read and replicated. And to do that, that necessitates DNA molecules being able to interact with each other and interlock very, very specifically. And that is actually mediated through hydrogen bonds. So some of you may be aware of the genetic code itself. The A, T, C, and G are actually sort of chemical elements. So they're called nucleobases. And here, what you see is G and C interacting through hydrogen bonds. So I said before, when talking about that, there's a non-carbon atom attached to a hydrogen. So this is a nitrogen connects to a hydrogen, which is hydrogen bonding to an oxygen. Similarly, we see here there's a nitrogen bonded to a hydrogen, covalent bonded, hydrogen bonding to a nitrogen. So that's a hydrogen bond, an intermolecular bond. So through this kind of specific sets of interactions, these nucleobases bond together and are then able to allow DNA to be read, translated, and replicated, allowing life to proceed. But these are not the only types of interactions that are really important within DNA. Within the 3-dimensional structure, these bases actually end up kind of stacked on top of each other. And if you look at them, you can see that actually these bases have a lot of carbon and hydrogen attach them. They look the kind of benzene rings. And these are actually able to stack on top of each other and interact via van der Waals forces, sort of cementing them together and creating a stable chain. Finally, there's also these negatively charged phosphates on the outside backbone of DNA. And as you recall, they're both negatively charged, so they're actually going to repel each other. And what this does is actually keep them far away from each other, keeping them on the outside of the molecule, and forcing the bases and protecting the bases which contain the information on the inside. So altogether, you have hydrogen bonds, which are locking the bases together. You have the van der Waals forces, which are gluing the bases down into the center of the DNA molecule. And you have the ionic interactions that are keeping the backbones apart and creating the double helix molecule that we all know and love. So now that we've sort of looked at how these forces exist in biology, let's go and translate these to how this works in protein drug interactions. So what I'm showing you here is a drug in a protein. And this is just a two-dimensional map. This is actually, it's not a drug, it's a hormone called norepinephrine. And what you have around it are different amino acids that have been highlighted from the protein. And I've sort of put little boxes around some of our favorite intermolecular interactions. So here you have an oxygen covalently bonded to a hydrogen, which is hydrogen bonding to this oxygen here. Here you have this carbon-rich ring, this benzene ring, which is interacting via van der Waals forces with this other benzene-like ring from an amino acid on the protein. Finally, you have this positively charged nitrogen on the drug, which is engaged in an ionic interaction with this negatively charged amino acid over here. So you can see that these interactions are actually sort of locking this drug into place in the protein. But as Shelley was describing, proteins aren't flat. They are actually, quite complex in terms of their three-dimensional architecture. So this two-dimensional map is not really useful for really representing how this works. So you can see over on the right, this is the same drug. It's norepinephrine bound to the same protein. And you see the protein represented by these ribbons that trace the amino acid backbone of the protein, with some of the amino acid structures highlighted. And here I've just sort of pointed out some of the same interactions that we see in the 2D map. So over here is the hydrogen bond. So you can see the covalent bond to the hydrogen, hydrogen bonding to this oxygen. And over here you have the positively charged nitrogen on norepinephrine, and with an ionic interaction to this negatively charged amino acid on the protein. But now that we're looking at this 3D structure, it sort of brings another question or another element of drugs interacting with proteins, that they have to interact in a 3D space. And you can see that they have to fit somewhere. So there's crevices or grooves or niches or all sorts of holes basically in a protein, and we call these pockets. And a drug needs to fit in that pocket, like a key in a lock. And so it's very important. So here you can see this is a psychoactive drug, which is interacting with a receptor in your brain. And you can see that it fits very nicely into this sort of hole in the protein, this pocket. And here's a cartoon of a different drug interacting with a different receptor in the brain. And you can see that it fits very nicely into this sort of crevice in the protein cross-section. If we zoom in on that, what we see actually is our three favorite intermolecular forces hard at work. So here there's actually an ionic interaction between the drug and an amino acid in the protein. We have some van der Waals forces, and we also have some hydrogen bonds as well. So what this is actually showing is not only does a drug need to be the correct shape to fit into a pocket in a protein, but it also needs to be able to position specific chemical elements in the right places to form strong intermolecular interactions with the protein itself. And this is a tall order. So if you're designing a drug, how do you go about doing this? How do you know how to build a drug in the right way to create those interactions? And the answer is really by understanding the three-dimensional nature of chemistry. And I'm going to go over that briefly. So this is perhaps the best-selling drug of all time. I think it actually is. It's called Lipitor, and it's actually used to treat or to prevent cardiovascular disease. And when we look at it in this two-dimensional form with the stick diagram, you know, of course, it looks flat. But when we sort of examine some of these structural features, we start to see some elements of why they might have been chosen by the people who designed this drug. So here, this sort of Y-shaped element that's composed of carbon and hydrogen, it obviously looks flat, but when you look at it in 3D, it sort of has this bent, almost pyramid shape. But if we look at this benzene ring down here, it is actually completely flat in 3 dimensions. Additionally, we can look at elements on the drug that may be serving to position certain pieces. So here, this is this oxygen connected to a carbon, connected to a nitrogen. This is called an amide bond. This is rigid and immobile. So that means it can't twist, it can't bend. So it places this part of the molecule completely static. It can't move. So if we compare that to this section of the molecule with this chain of carbons with these oxygens coming off of it, this is actually rotatable and floppy. And when we look at it in 3D, we can actually see that that whole chain is kind of bent and twisted at the same time. So why would it be designed this way? Why does this molecule look like this? And why have these specific structural elements been chosen? So I'm going to show you on this slide that it's interacting with its target, which is an enzyme called HMG-CoA reductase. And here I've just pointed out some of the elements on this molecule that can perform some of our intermolecular interactions. So this element here that I said was mostly carbon can perform van der Waals interactions. These oxygens bonded to hydrogens are able to perform hydrogen bonding. And this part of the molecule here can be negatively charged and engage in ionic interactions. So then when we take this structure and visualize it inside its protein pocket in the active site, as Shelley was describing, we can see that its 3-dimensional structure positions each of these elements in a very specific place to interact with the protein in a very specific way to build these intermolecular interactions. But why is it interacting this way? What is it doing in this protein pocket? Well, it's interacting specifically with HMG-CoA reductase to actually shut down its function, to stop it from working and block its activity. So why do we care? What does that do? Well, it's actually stopping the biosynthesis of cholesterol and also telling your cells indirectly to pull in and extract cholesterol out of your blood. So what happens when you do that? Well, you're reducing arterial plaque buildup, meaning that you get less sort of fat being deposited on the inside of your blood vessels, which then lowers your blood pressure. And finally, what's that going to do to your health? Well, it's reducing your risk of cardiovascular disease and maybe preventing you from getting a stroke or a heart attack. So today what I've wanted to tell you is that drugs are interacting with proteins to change biology. And the way that they do that is through these intermolecular non-covalent interactions, which come in 3 types: ionic, hydrogen bonds, and van der Waals forces. In addition to this, drugs need to be fit to a 3-dimensional pocket in a protein. And in order to be successful, they need to be able to also fit in the pocket and form these interactions with the protein. And the way that you do this, if you're designing a drug, is to use the 3-dimensional nature of chemistry to design something that orients the right chemical groups into the protein pocket to form a perfect set of interactions to interact specifically with a therapeutic target of interest and to change biology in a way that affects human health. So thank you for listening to our talk today. Stay tuned. Next week, Vijay Shahani and David Cuter are going to be taking you on sort of the next step of sort of the drug development process and talking about drug development fundamentals and computer-aided drug design. Thank you very much.
Thank you very much, Ben and Shelley. A fantastic presentation of highlighting the elements of biology and chemistry and how that influences diseases and how we can use drugs to kind of treat those diseases. So we do have a few questions that we'll be answering here. The first question is to Ben. Regarding the slide that shows hydrogen bonding, can X be any element except carbon?
It's a very good question. And the answer is not any element. I think generally in biology, there's certain elements that are used. So generally you'll see that as being oxygen, nitrogen, sulfur, sometimes phosphorus. But in terms of the general schematic of chemistry, the answer is no. In terms of biology, there's the specific elements that are not carbon. Yeah, it has to do with electronegativity. That's the sort of fundamental driving force.
Great. Our next question highlights sort of what do drugs interact with? Is it just proteins, or are there other elements within the cell that a drug can interact with?
Well, that's also a fantastic question. And the answer is there are lots of other things that drugs can interact with in the cell. Sort of a very, the traditional paradigm in drug discovery is sort of protein, of drugs that bind to proteins, but especially now we're seeing that we can find drugs that interact with nucleic acids, with sugars. Even there's drugs that interact with lipids in the cell wall, or cell membrane. So the answer is no, but generally the most traditional view is that they do interact with proteins.
Great. This next question is to Shelley. In the example that you gave for cancer, could drugs interact with also healthy receptors?
That's a really great question. So sometimes when we're targeting elements that, you know, our normal cells do have, sometimes this does result in some unwanted side effects. And because as we said, we're targeting something that can also be on some healthy cells. So that can sometimes lead to those, you know, undesirable effects. But in the case of, in the example we gave in terms of receptors, because the cancerous cells have so much more of them, we can target them a little bit more specifically. So yeah, there is an element that some of your normal cells can also be affected.
Great. The next question is to Ben. Regarding sort of the molecular shapes that you were discussing, why is it that certain shapes are flat, and why is it that certain shapes can be kind of bent?
That's a good question. All of these questions are very good and are things that I didn't want to talk about because they can get complicated. But the answer actually is the way that it's a concept called hybridization. And I'm not going to get into it too much, but it means that when certain atoms like to adopt certain geometries, and that causes certain functionalities to have different sort of positions. And that is another sort of element of bonding that I didn't talk about, and another sort of element of drug design that you can also sort of layer on top. But the answer to that is hybridization, if you want to look it up.
Yeah, it's definitely a more complex topic. In terms of other questions that we have here, yes, these sessions are being recorded and they will be shared on our YouTube channel. So stay tuned for that. Let's see if we have any other questions here. In terms of, I guess, subsequent topics we will be covering, they will be announced on a weekly basis. We will be sort of discussing machine learning, AI, and how those sort of tools fit within the drug discovery pipeline and paradigm. So stay tuned for those more advanced topics. Otherwise, I think that is all the questions that we have for today, unless any stragglers want to ask any more questions. But I hope this session sort of provided a high-level overview of, again, the biology and the chemistry behind diseases and designing drugs. In our next talk, we will be sort of going over what are the specifics for drug development and how computer-aided drug design and computational tools can be used to assist the development of medicines. Oh, we actually have one more question coming here. Is gene therapy considered a better alternative in comparison to drugs? For example, editing a cancer gene versus using medicine, which I'm going to guess is small molecule.
I can answer that, and I think that it's something that I think there's a lot of interest in currently. The answer to that really lies in the way that these therapies are delivered. Right now, we know a lot. maybe not enough, but we know a lot about how drugs are absorbed and metabolized and changed by the body, and how do they get to their target. For gene therapies, the safety of that and the timing and all of that is just not as well known and is not as well sort of characterized. So certainly in terms of efficacy, you could definitely imagine that that's probably better. But in terms of safety and in terms of, you know, how easy it is to take for just a regular person, the answer is probably it's not as easy right now. But I think that's really rapidly changing as we learn more about what gene therapy is, how it works, and how to deliver these kinds of medicines to people.
Great, and I think you touched on something really important is that, you know, when you have a disease or sort of, you know, a problem that you're trying to target, you sort of need to use the best, you know, tool that you have to treat those. And then, you know, physicians and groups of scientists kind of work together to really understand what's happening at the biological level and what tools can we use, you know, to solve this and make the lives of patients better. Great.
Actually, just chiming in a little bit on the last question, there was some recent news from multiple groups that published for the gene editing, in that there's a lot of unanticipated, to speak to Ben's point, there's a lot we don't know. And it was just a very recent finding in the last few weeks that it's actually modifying parts of your DNA that was not intended to in any shape or form. And so those can have large impact as well. So as we learn more about, you know, these targeted therapies and their limitations, you know, it's still very novel. And this is just a really recent hot topic example of how gene editing is really in its infancy to a large extent.
Great. I think if we don't have any more questions, we will end the webinar here. We thank everyone for attending. We will be following up again with the recording for this session and future sessions. So please stay tuned, and the topics will be announced on our LinkedIn and Twitter page. Thank you all for attending.